morning, everyone. My name is Chauhan, and I'm working as an assistant professor in IIMT College of Pharmacy, Greater Noida. So today I'll be delivering a video lecture on the topic of proteins. Before that, we should know what are amino acids. Amino acids are a group of organic molecules that consist of a basic amino group and an acidic carboxyl group, COOS, an organic R group that is unique to each amino acid. Basically, as you can see in the structure right below, each molecule contains a central carbon atom called alpha carbon to which both an amino and carboxyl group is attached. The remaining two bonds of the alpha carbon atom are generally satisfied by a hydrogen atom and an R. So let's look up in the examples. These are the various type of amino acid with R substitution of different groups. So let's talk about alanine. The R substitution is of methyl. In valine, the R substitution is of isopropyl. And leucine, the R substitution is of isobutyl. In glycine, the R substitution of hydrogen. And if we took an example of a benzene ring containing amino acid, this is the substitution of benzyl group in alanine. So this is what we call phenylalanine. So let's move on the classifications. There are basically two types of classification. First, based on chemical nature. And the second is based on the nutritional requirements. So if we talk about based on chemical nature, it is basically the amino acid have various organic group are besides amino and COS group. They are classified according to this group. Naturally occurring amino acids are categorized into three main groups. First, aliphatic, aromatic, and heterocyclic. If we talk about an aliphatic, there are four different types of amino acids classification. First, we should know what are aliphatics. Amino acid, according to nature, they are classified in the basis of natural. Natural, they have same amount of acetic and alkaline group. They have only one carboxyl and one amino group in its structure. Examples: glycine, valine, and serine. Next, if we talk about acidic group, these have two carboxyl group and one amino group in its structure. They have acidic nature due to extra carboxyl group. Third is alkaline. They have an extra amino group at R position. So they are alkaline in nature. Last, amino acid with sulfur, basically name it's told that there is a presence of sulfur group in its structure. Example are cysteine, methionine. Okay. Move on the next classification, which is aromatic. Aromatic amino acids are these amino acids have benzene ring with the R position. Next, heterocyclic amino acid, they have heterocyclic molecules in the lateral organic groups. So, I've already told you about this. So, next classification is based on nutritional requirements, essential and non-essential. So, essential amino acids are basically the amino acid which cannot be synthesized by the body and therefore need to be supplied through the diet. Non-essentials are which body can synthesize to meet biological needs. So let's learn these amino acids in a fun way. There are basically 10 essential and non-essential amino acids, in which the first, if we talk about the essential, there is a trick, goes like VIP, call me PT. So the amino acids will be valine, VIP, V, valine, isoleucine, phenylalanine, Hol, histidine, arginine, lysine, leucine, and methionine, TT is threonine and tryptophan. This will be easy to learn. So next is non-essential. Is almost all girls go crazy after getting tasty potato samosa. So the trick is like alanine, almost, and asparagine, glycine, glutamine, cysteine, aspartate, glutamate, tyrosine, proline, serine. Next is biological rules of amino acid. Amino acids serve as the building blocks of proteins. Some amino acids are found in free form in human blood. They also serve as precursor of hormones, purine, pyrimidine, vitamins, and biological important amines like histamine. Next is peptides have many important biological functions. Some of them are hormones. They are used as antibiotics, anti-tumor agents. Some peptides are required for detoxification reactions. Some peptides serve as neurotransmitters. Amino acid protein protects living organelles against free radical induced damage. Some peptides are involved in regulation of cell cycle and apoptosis. So now we know what are amino acids. Next we will move on proteins. What are proteins? Proteins are the polymer of amino acid and they are connected to each other by a peptide bond or peptide linkage. Chemically, peptide linkage is an amide bond between the carboxyl group and amino group. So the reaction between two molecules of amino acid proceeds through the combination of amino group of one molecule with the carboxyl group of the other. I'll take the example right here. For example, when the carboxyl group of glycine right here and combines with the amino group of an alanine, right? So it will form peptide linkage and called glycyl alanine. Now the product of the reaction is called dipeptide because it is made of two amino acids, right? When the number of such amino acids, now we will talk about polypeptide and protein. Okay, we should clear that concept first. When the number of such amino acids is more than 10, then the products are called polypeptides. Okay, now the question arises is when can a polypeptide be called as a protein? Polypeptides which have a molecule mass of 10,000 Dalton or more are called protein. Basically, a polypeptide with more than 100 amino acid residue having a molecular mass higher than 10,000 Dalton is called a protein. However, the distinction between a polypeptide and a protein is not very sharp. Polypeptides with fewer amino acids likely to be called proteins, such as you can take an example of insulin. It contains 51 amino acids in its structure. Now, if we talk about the structure of protein, they are basically divided into two parts: primary, secondary, tertiary, and quaternary. So we'll first talk about primary structure of protein. The sequence of amino acids in the peptide chain is called the primary structure. Primary structure, in primary structure, we'll talk about only the sequence of amino acids, right? Any change in the primary structure, that is the sequence of amino acid, or creates a different protein. Then we will see here in the next slide, there is a structure of an amino acid sequence, right? So the free alpha amino group written to the left is called the amino terminal or N-terminal N. The free alpha carboxyl group written to the right is called the carboxyl terminal or C-terminal N. Okay, this will be easy. Next, there is a secondary structure of protein. The next level of protein structure, secondary structure, refers to local folded structure that form with a polypeptide due to interaction between atoms of the backbone. Backbone will be like that. These structures rise due to the regular folding of backbone of the polypeptide chain due to hydrogen bonding between the amino group of peptide bonds. There will be a hydrogen bonding of the peptide backbone causes the amino acid to fold into a repeating pattern. There will be two shapes. The first is plated sheets like paper folding, and next is alpha helix, this spiral structure. Okay, so these are the structure. This is the beta plated sheets, and this is alpha helix. The amino group of the amino hydrogen backbone are connected with each other. Next is tertiary structure of protein. The overall three-dimensional structure of polypeptide is called its tertiary structure. The tertiary structure is primarily due to interaction between the R group of the amino acid that makes up the protein and hydrophobic R group clustered together on the inside of the protein. Okay, right here. The hydrophobic R group will be inside here and the hydrophilic amino acid will be the outside to interact with surrounding water molecules. So Hydrophobic interaction in which amino acid with non-polar hydrophobic ion group cluster together on the inside of protein and leaving hydrophilic amino acid on the outside to interact with surrounding water molecules. So, last is quaternary structure of protein. Many proteins are made up of single polypeptide chain and have only 3 levels of structure, the one we have already discussed. However, some proteins are made up of multiple polypeptides. Polypeptides are containing 10 amino acids in its structure, right? So there will be a multiple polypeptide chain and will be in a cluster form. Subunits came together to give the protein its quaternary structure. So I hope this part must be clear, right? Look in this structure right here. Let's take a review. This is the polypeptide chain, amino sequence, primary. Secondary is alpha helix, beta plated sheet, will be in 2 forms. Third is tertiary structure, hydrophobic. Due to hydrophobic nature, they will be in the inside and the outside. The hydrophobic part will be inside, and the hydrophilic part of the amino acid will be outside. Last is quaternary structure. The subunit 1 and subunit 2 polypeptide chain will be in a cluster. If we talk about the classifications, there are 3 basic classifications: simple, conjugated, and derived. Simple, what are simple proteins? Simple, they give only amino acid or their derivative on hydrolysis. Globular will be, they have a spherical shape and are soluble in water, acid and bases. Fibrous are, these are long strand like protein that are insoluble in water. If we talk about conjugate, it contains non-protein group or compound in its structure containing amino acid residue attached to a protein part. What are simple? Simple are just containing amino acid in its structure. In conjugated, there will be another non-protein group attached to the structure in the protein. So what will be? These are nucleoproteins. It contains nucleic acid, right? This is glycoprotein. It contains glycogen. This is lipoprotein. It contains lipid. Phosphoprotein is phosphorus group, right? So these are the examples. And what will be the derived group? Derived protein are degradation products obtained by the action of acids, alkalis, and enzymes on protein. So these are the classification. If we talk about the biological importance of protein, plasma membrane proteins regulate the transfer of many substances across the cell membrane. All receptors are protein in nature. We already know that. All enzymes are protein in nature. All antibodies are protein in nature. Immunoglobulins. Some hormones are protein in nature, like insulin, which is and growth hormone. Okay. Next, some protein are protective. Example, keratins will be found in skin, hair, and nails. Make the skin resistant to chemical. Some protein are supportive. Example, collagen, the most abundant protein in animals. Hemoglobin is protein, carries oxygen in blood through all over the body. Actin and myosin are contractile protein found in muscle cells and responsible for muscular contraction. So this is the importance and function. If we talk about the function, first is catalytic function. If we talk about the enzymes, what they do. All chemical reactions in biological system are catalyzed by specific enzymes. Second function will be transport and storage. Hemoglobin transport oxygen, right? Myoglobin carries stores oxygen in muscle. Albumin transport free fatty acid in blood. Transferrin transport iron in blood. Okay, so these are the functions. If we talk about defense function, clotting factor prevent loss of blood. Immunoglobulins protect against the infection. So these are the qualitative tests for protein. How we know that this liquid, this compound contains amino acid or protein. So these are the tests. First will be Nin-Hydrate test, which is very famous. The amino group belongs to a free amino acid undergoes a chemical reaction, the Nin-Hydrate, which behaves as an oxidizing agent. Now, how do we perform it? First, a 2% solution of anhydride must be prepared by dissolving 0.2 gram of anhydride in 10 ml of either ethanol or acetone. Now, 1% solution of amino acid in distilled water must be prepared. A few drops of 2% anhydride solution must be added to the solution. The test tube must be kept in a warm water bath for approximate 5 minutes. The development of a deep blue violet thread indicates the presence of amino acids. So look in the image right here. This shows the positive result and that will show the negative. Next test will be Biuret test. Take 3 clean and dry test tubes. Add 1 to 2 ml of test solution. Ag albumin. We will compare. Okay, test in standard. Test solution, ag albumin and deionized water. You will take 3 test tubes containing these solutions in a respective test tubes. Add 1 to 2 ml of Biuret reagent to all the test tubes. Shake well and allow the mixture to stand for 5 minutes. Observe for any color. So, this is the sample. If it shows blue color, the Biuret test will be negative. If it shows purple color, the Biuret test will be positive. Next is xanthoproteic test. The xanthoproteic test is a biochemical test for the detection of amino acid containing phenolic or indole group. This is specific test, right? Phenylalanine to find out tyrosine and tryptophan, aromatic amino acids. So how do we perform it? About 1 ml of sample solution is taken in test tube. To this, the same amount of concentrated nitric acid is added. The test tube is allowed to cool down to room temperature. If the sample is protein solution, a white PPT might develop due to denaturation of protein. Then 1 ml of 40% NaOH solution is added to the So, this is a xanthoproteic test definition principle procedure result chooses. So, next will be Hopkin-Cole test. In this test, take a test tube and add 2 ml of light exposed glacial acetic acid and 2 ml of sample liquid are taken. To this, concentrated acetylcholine is added along the sides of test tube held at slanting position. The test tube should be observed for the formation of coloring at the interface of 2 layers. Next is Millen's test. Millen test is an analytical test used for the detection of amino acid tyrosine, which is the only amino acid containing the phenol group. Principle procedure. About 2 ml of sample solution or the first 1% tyrosine solution is taken in a test tube. To this, about 2 ml of Millen's reagent is added. The test tube are then kept in water bath for about 2 minutes. If a red colored precipitate is not observed immediately, the tubes are then observed for the formation of colored precipitate. So, for identifying the phenolic proteins, it shows pink or red color in a test tube. If we talk about Sakaguchi test, it is used as a qualitative test for arginine that is either free or in protein. The test was discovered by named after the Japanese scientist Shoyo Sakaguchi in 1925. About 3 ml of test solution is added to a test tube, to which 1 ml of 40% NaOH is added and mixed correctly. Then 2 drops of naphthol are added to the same test tube and mixed through. Now 4-5 drops of 10% sodium hypochromite or bromine water are added. The test tube is observed for the development of color. So now what are the problems that arises in malnutrition of proteins? There are 2 diseases which are very dangerous. First is kwashiorkor, also known as edematous malnutrition because of its association with edema. There will be a swelling in liver, right? So it will be shown in example right here. This is normal body, and this is the body of a kwashiorkor person having a kwashiorkor. Due to lack of protein or severe malnutrition in the body, this problem arises. So it is a form of malnutrition caused by lack of protein in the diet. People who have kwashiorkor typically have an extremely emaciated appearance in all body parts except their ankles, feet, belly, which swell with fluid. What causes kwashiorkor is caused by lack of protein in the diet. Protein is also especially important for growth during childhood and pregnancy. So, what are the symptoms? Change in skin hair color, fatigue, diarrhea, loss of muscle mass, failure to grow or gain weight, edema, flaky rash, shock. How is kwashiorkor diagnosis? The test may be performed in blood, urine to measure sign of malnutrition and lack of proteins. So treatment is very easy to take proper amount of protein in the diet, more calories overall, especially if treatment is started early. You may first be given more calories in the form of carbohydrates, sugar, and fats. Once these calories provide energy, you will be given food with protein. The next malnutrition disease is marasmus. Marasmus is a form of severe malnutrition. You can see right here, differentiate between these images. Kwashiorkor here. You can see my cursor. This is kwashiorkor, right? And this is marasmus. There will be a bulging of abdomen, moon face, thin and brittle hair. But in marasmus, thin and brittle hair also present, but with a cement face and loss of muscle mass. It is an extreme severe type of nutrition disorder in which there is a significant wasting of fat, muscle, and tissue of the body. So causes, nutrient deficiency is the main cause of marasmus. Obviously, it occurs in children that don't ingest enough protein, calorie, carbohydrate, and other important nutrients. This is usually due to poverty and scarcity of food. There are several types of malnutrition. A malnourished child may have something other than marasmus, iron, iodine, zinc, vitamin E. Right? How we can diagnose that? A doctor can often make a preliminary diagnosis of marasmus through a physical exam. A lack of motion in malnourished child may also help confirm a diagnosis of marasmus. So, how is marasmus treated? Initial treatment of marasmus often include dried skim milk powder mixed with boiled water. Later, the mixture can also include vegetable oils such as sesame, casein, and sugar. So once the recovery started, they should have more balanced diet than meets their nutritional needs. If dehydration is a problem because of diarrhea, rehydration should also be tried. It should take more liquid diets, right? A child may not necessarily need fluids delivery intravenously. Oral hydration may be sufficient. Thank you so much for patiently listening to my video lecture. Have a nice day.